Okay, good morning, class. Our topic for today's lecture is on male reproductive endocrinology. This is just a continuation of our topic last meeting since we already discussed on the introduction or overview of reproductive endocrinology. But just to have a quick recap, we discuss on male gross anatomy, spermatogenesis, testicular descent and thermoregulation, and we also talk about erection and ejaculation. For today's lecture, we will be discussing on endocrine function of the testis, blood-testis barrier, secondary characteristics of male, sperm anatomy and physiology. Some of the topics are have already been discussed in your other subjects. So this is some of the topics are already just a part of your review. Okay. So to begin with, let me show you the chemistry and biosynthesis of testosterone. So I would just like you to know how testosterone is produced from cholesterol. So, testosterone is the principal hormone of the testis, and it is a C19 steroid with a hydroxyl group in the 17th position, as you can see in the figure. So, it is synthesized from cholesterol in the Leydig cells, and it is also formed from androstenedione secreted by the adrenal cortex. So the biosynthetic pathways in all endocrine organs that form steroid hormones are similar. The organs differ the organs differing only in the enzyme systems that they contain. So in this figure, it also shows how this dehydroepiandrosterone and androstenedione is being produced from the primary cholesterol. Okay, so in the Leydig cells, the 11 and 21 hydroxylase are found in the adrenal cortex. But here, it is absent. The only present here is the 17A hydroxylase, which you can see above. And the pregnenolone is therefore hydroxylated in the 17th position and then subjected to the side chain cleavage to form this dehydroepiandrosterone. So androstenedione is also formed via progesterone and 17-hydroxyprogesterone. But in this pathway, the pathway of this androstenedione is less prominent in human. So dehydroepiandrosterone and androstenedione are then converted to testosterone. The secretion of this testosterone is under control of luteinizing hormone and mechanism by which luteinizing hormone stimulates the Leydig cells also involves increased formation of CAMP via the G protein coupled LH receptor and the GS. So cyclic AMP increases the formation of cholesterol from cholesterol esters and the conversion of cholesterol to pregnenolone via activation of protein kinase A. So protein kinase A is a type of enzyme. So let me show you now the endocrine function of this endocrine function of the testis. So first is secretion. The testosterone secretion rate is 4 to 9 milligram per unit. I think this is deciliter. Or it is approximately 13.9 to 31.33 nanomole per unit in normal adult males. So small amounts of testosterone are also secreted in females, with the major source being the ovary, but possibly from the adrenal as well. So in this table, you can see that 98% of the testosterone in plasma is bound to protein. 65% is bound to beta globulin called gonadal steroidal binding globulin, or we call this GBG, or sex steroid binding globulins, and 30% to the albumin, 30% only to the albumin. So GBG also binds estradiol. The plasma testosterone level, or the free and bound, is about 300 to 1000 nanogram per deciliter in adult men. Okay, so compared with the 30 to 70 nanogram deciliter in adult women. So it declines somewhat with age in males. So in this figure, it shows that a small amount of circulating testosterone is converted to estradiol, but most of the testosterone is converted to 17 ketosteroids, principally androsterone and its isomer et ethylcholanolone. and excreted in the urine. So about two-thirds of the urinary 17 ketosteroids are of adrenal origin, and one-third are of the testicular origin. Although most of the 17 ketosteroids are weak androgens, they have 20% or less the potency of testosterone. It is worth emphasizing that not all 17 ketosteroids are androgens, and not all androgens are 17 ketosteroids. Ethioholanolone, for example, has no adrenergic activity, and testosterone itself is not a 17 ketosteroids. Okay, since this is already a adrenal origin. So next is the actions. In addition to their actions during development, testosterone and other androgens exert an inhibitory feedback effect on pituitary luteinizing hormone secretion. Remember what I have shown you last meeting? So I've shown you a figure or a diagram on this about this inhibitory feedback effect. So other action is to develop and maintain male secondary sex characteristics. And the third is exert an important protein anabolic growth promoting effect. And lastly, along with FSH, it maintains spermatogenesis. So remember the highlighted text na lang. Okay. So ito yung this one is shown on the first slide of this lecture. Okay. So here, FSH is a tropic for Sertoli cells, and follicle-stimulating hormones and androgens maintain the gametogenic function of the testis. FSH also stimulates the secretion of ABP and inhibin. Okay, and inhibin feedback to inhibits FSH secretion. Luteinizing hormone is tropic for Leydig cells, and it stimulates the secretion of testosterone, which in turn feeds back to inhibit the LH secretion. So hypothalamic lesions in animals and hypothalamic disease in humans lead to atrophy of the testis and loss of their function. So when this hypothalamus is injured during certain activity, or you have these hypothalamic lesions, it will lead to the atrophy of the testes and loss of their function, since these other hormones are also affected. Okay, again, um, I repeat, this is a figure of postulated interrelationship between the hypothalamus, anterior pituitary, and the testis. So the solid arrows indicate the excitatory effect, whereas the dash arrow indicate the inhibitory effect. Don't forget that, ha? Since yung solid, siya yung solid arrow, it shows the excitatory. Yung mga nag-inhibit naman going to the, inhibit the secretion of hormones going to the hypothalamus and vice versa are those inhibitory effects. Okay. Castration is followed by a rise in the pituitary content and secretion of FSH and LH, and hypothalamic lesions prevent the rise. Testosterone inhibits the LH secretion by acting directly on the anterior pituitary and by inhibiting the secretion of this GnRH from the hypothalamus. Inhibin acts directly on the anterior pituitary to inhibit FSH secretion. In response to LH, some of the testosterone secreted from the Leydig cells bathes the seminiferous epithelium and provide a high local concentration of androgen to the Sertoli cell that is necessary for normal spermatogenesis. Okay, so let's now proceed with the secondary sex characteristics of male. So this table shows the changes at puberty in boys, basically the male secondary sex characteristics. So the widespread changes in hair distribution, body configuration, and genital size that develop in the boys at puberty are some of the male secondary sex characteristics as summarized in this table. So you can see that there's increase in penis length and width, scrotum becomes pigmented and rugose. So for the internal genitalia, we have seminal vesicles enlarged, and it secretes and begins to form the uh, fructose. The prostate and seminal vesicles enlarge, and the seminal vesicles begin to secrete also fructose. So this is a sugar that appears to function as the main nutrition supply for the spermatozoa. The psychic effects of testosterone are difficult to define in humans, but in experimental animals, androgens provoke boisterous and aggressive play. So, for the other, you can just read other secondary sex characteristics like in boys, hair growth, mental body conformation, and the skin. So although body hair is increased by androgen, scalp hair is decreased, and hereditary baldness often fails to develop unless dihydrotestosterone is present or the DHT is present here. So yung testosterone pala can affect the hairline of usually male. Okay. So in assessing a physical examination also, some of this male sexual development are observed. So this is called Tanner stage. We have also Tanner stage for breast, but this is Tanner stages of sexual development of male reproductive system from stage 1 to stage 5. Okay, you can just go back to this table na lang to see or read what description or what happens to the male reproductive system during each stages from stage 1 to stage 5. Okay, so proceeding to the blood testis barrier, the walls of the seminars in this figure, you can see that the walls of the seminiferous tubules are lined by primitive germ cells and Sertoli cells. Okay, there are Sertoli cells here and primitive germ cells. They are also lined by complex glycogen containing cells that stretch from the basal lamina of the tubule of the lumen. Here's the basal lamina. Okay. Germ cells must stay in contact with Sertoli cells to survive. This contact is maintained by these cytoplasmic bridges. So, parang may mga frame sila to maintain the contact. So, tight junctions between adjacent Sertoli cells near the basal lamina form a blood testis barrier that prevents many large molecules from passing from the intestinal tissue and a part of the tubule near the basal lamina to the region near the tubule lumen. However, steroids penetrate this barrier with ease, and evidence suggests that some proteins also pass from Sertoli cells to the Leydig cells and vice versa to function in a paracrine fashion. In addition, maturing germ cells must pass through the barrier as they move to the lumen. This appear to occur without disruption of the barrier by a coordinated breakdown of tight junctions above germ cells and formation of new tight junctions below them. So the fluid in the lumen of the seminiferous tubules is quite different from plasma because it contains very little protein and glucose, but is rich in androgen, estrogen, potassium, inositol, and glutamic acid and aspartic acid. Maintenance of its compositions depend on the blood testis barrier as well. So this barrier also protects the germ cells from blood-borne noxious agents and prevents antigenic products of germ cell division and maturation from entering circulation and generating an autoimmune response, and may establish an osmotic gradient that facilitates movements of fluid into the tubular lumen. Okay, sorry, a wrong slide. Just have to proceed with the next slide. Okay, so just for a quick review, spermatogonia, this is the primitive germ cells next to the basal lamina, as well as you can see in the figure. And this spermatogonia mature into primary spermatocytes. So we have been discussing this last time or last meeting But then I'm just going to show you a quick review or a quick short video of this.
Sperm develop in the seminiferous tubules of the testes. Primordial sperm cells, called spermatogonia, lie dormant in the outer compartment of the tubules from the late fetal period until puberty. At puberty, the spermatogonia undergo mitotic divisions, giving rise to genetically identical cells. The cells grow gradually into primary spermatocytes, preparing to divide into cells half their size. Each of these primary spermatocytes undergoes a first meiotic division, forming two smaller haploid cells known as secondary spermatocytes. These secondary spermatocytes divide once more, giving rise to four haploid cells called spermatids, containing exactly half of the father's genetic material. Through a process known as spermiogenesis, the spermatids change shape, transforming into mature sperm cells. The spermatid's nucleus condenses, the acrosome appears, and the tail begins to grow. Much of the cytoplasm is shed. As spermiogenesis completes, the sperm cells break free from the Sertoli cell lining of the tubules into the lumina. The sperm cells are then stored in the epididymis, where they functionally mature.
Okay, I hope the short clip is clear since it discusses all of our discussion last. I mean, regarding the spermatogenesis, which we already discussed. Okay, so this shows the cross section through seminiferous tubules. Okay, so you can see the lumen here, the Sertoli cells, the spermatogonium, and then the Leydig cells. So this basal lamina protects this lumen. Okay, and also they have undergone mitosis and the first and second meiotic division in order for the spermatogonium to become a spermatozoa or the sperm itself. Okay. So we already discussed this one, the period of, I mean, steps on spermatogenesis. We have period of multiplication, period of maturation, and differentiation. And also we have just a short review of this one. We already discussed this as well. So this process begins during adolescence. The primary spermatocytes undergo meiotic division, reducing the number of chromosomes, and in this two-stage process they divide into secondary spermatocytes and then into spermatids, which contains the haploid number of 23 chromosomes. Next is the spermatids mature into spermatozoa, or what we call the sperm, and as a single spermatogonium divides and matures, its descendants remain tied together by cytoplasmic bridges until the late spermatid stage. This arrangement helps to ensure the synchrony of the differentiation of each clone of germ cell. So the estimated number of spermatids formed from a single spermatogonium is 512, whereas the formation of a mature sperm from a primitive germ cell by spermatogenesis in human spans approximately is 74 or approximately 74 days. Okay. So let's now proceed with the sperm anatomy and a little bit of physiology. So each sperm is intricated by a motile cell, which is rich in DNA. So yung ano niya is rich in DNA and with a head that is made up mostly of chromosomal material. Covering the head like a cup is the acrosome. We call this as macrosome. And the lysosome-like organelle rich. This is an lysosome-like organelle rich in enzymes involved in sperm penetration of the ovum and other events associated with fertilization. You can also recall the videos that I presented last time. Okay, the motory tail of the sperm is wrapped in its proximal portion by a sheath holding numerous mitochondria. So, yung mitochondria are located in the tail of the sperm. The membranes of the late spermatids and spermatozoa contain a special small form of angiotensin converting enzyme called the germinal angiotensin converting enzyme, or we call this as GACE, GACE. This GACE is transcribed from same gene as the somatic ACE. However, this GACE displays tissue-specific expression based on alternative transcription initiation site and alternating splicing patterns. So the full function of this enzyme GACE has yet to be elucidated, although GACE specific knockout mouse models are sterile. So then spermatids mature into spermatozoa in deep folds of the cytoplasm of the Sertoli cells. Mature spermatozoa are then released from Sertoli cells and become free in the lumens of the tubule. The Sertoli cells secrete androgen binding ABP, or we call this ABP, inhibin, and the MIS, or this MIS is the Mullerian inhibiting substance. So they do not synthesize androgen, but they contain aromatase, this CYP19. So this CYP19, or the aromatase, is the enzyme responsible for conversion of androgens to estrogen. Again, I repeat, this CYP19 is the enzyme responsible for the conversion of androgen to estrogen, and they can produce estrogen. Okay, so this one produces estrogen. So the ABP, or the androgen binding protein, functions to maintain a high stable supply of androgen in the tubular fluid, and inhibin inhibits follicle stimulating hormone secretion, or FSH. So FSH and androgens maintain the gametogenic function of the testis. So after when you do hypophysectomy, injection of this luteinizing hormone produces a high local concentration of androgen in the testes, and this maintains spermatogenesis. So the stages from spermatogonia to spermatids appear to be androgen independent, so they do not depend on androgen. However, the maturation from spermatids to spermatozoa depends on androgen acting on the Sertoli cells, in which the developing spermatozoa are embedded. FSH, or the follicle stimulating hormone, acts on the Sertoli cells to facilitate the last stage of spermatid maturation. In addition to, it promotes the production of androgen binding protein. So here, an interesting observation is that the estrogen content of the fluid in the reed testes, you can locate the reed testes here, is high, and the walls of the reed testes contain numerous alpha estrogen receptors, or we call this as ERA or ERA. In this region, fluid is absorbed and the spermatozoa are concentrated. If this does not occur, the sperm entering the epididymis are diluted in a large volume of fluid, and then this will result to infertility or reduced fertility. Okay, so let's proceed now to the further development of the spermatozoa. Spermatozoa then leaves the testes are not fully mobile, or meaning to say they are immobile. Then they continue their maturation and acquire motility during their passage through the epididymis. So, motility is obviously important in vivo, meaning to say inside. But fertilization also occurs in vitro outside if an immotile spermatozoan from the head of the epididymis is microinjected directly into an ovum. That's why we have this in vitro fertilization. So the ability to move forward, or progressive motility, which is acquired in the epididymis, involves the activation of a unique set of proteins from the cat spur family. Okay, so this cat spur family are, it's a protein which are localized to the principal piece of the sperm cell. Remember that this is a cat spur. Okay, cat spur. So this cat spur form an alkaline sensitive calcium 2 channel that becomes more active as the sperm go from the acidic vagina. The acidic vagina, it contains pH that is 5, to the cervical mucus, which is pH of 8. So sperm from knockout mice that do not express this Caspr 1 to 4 have altered motility and are infertile, emphasizing the important role of this protein, this Caspr. So in addition to spermatozoa also express olfactory receptors, and ovaries produce odorant-like molecules. Recent evidence that these molecules and their receptors interact, fostering movement of the spermatozoa towards the ovary. So this we call this as chemotaxis. Okay. Next is, so this is now the flow. I mean the stages also. So ejaculation of the spermatozoa involves this contraction of the vas deferens, mediated in part by P2X receptors, ligand-gated cation channel that respond to ATP. And fertility is reduced in mice in which these receptors are knocked out. So then, once ejaculated into the female, the spermatozoa move up to the uterus of the isthmus of the uterine tubes, where they slow down and undergo capacitation. Okay, so this further maturation process involves two components for maturation. We have this increasing the motility of the spermatozoa. And number two is facilitating their preparation for the acrosome reaction. So that, however, the role of capacitation appears to be facilitatory rather than obligatory, because fertilization is readily produced in vitro. From the isthmus, the capacitated spermatozoa move rapidly to the tubal ampullas where fertilization takes place. Okay, remember this one because uh, will be discussed also in your future lectures. So that's the in ampulla, it is the location or the area which fertilization takes place in the female organ. Okay, so we're now heading to the composition of human semen. So in this table, you can see that the fluid is ejaculated at the time of orgasm. The semen contains sperm and the secretion of the seminal vesicles, prostate, Cowper's gland, and probably the urethral glands. So we can see that the color is white, the specific gravity is 1.028, pH is about 7.35 to 7.50. So there's an average volume per ejaculate that is 2.5 to 3.5 ml after several days of abstinence from sexual activity. So the volume of semen and the sperm count decrease rapidly with repeated ejaculation. Even though it takes only one sperm to fertilize the ovum, each milliliter of semen normally contains about 100 million sperm cell or sperm. So reduction in sperm production is associated also with infertility, that 50% of men with counts of 20 to 40 million per milliliter, and essentially all of those with counts under 20 million per ml are sterile. So this is just to show you the some of the sperm morphologies. So the presence of many morphologically abnormal or immotile spermatozoa also correlates with infertility. As you can see, only one here is one out of 15. Only one survives. This is the normal. Okay, so other are abnormal. You can see that there's a microsperm, megalosperm with a big head, microsperm head with a small head, roughened head membrane, elongated forepart. Okay, worst is this 8 and 9, which is absent body, or we have this arrested development of tail, no tail or no body. Okay. Also, we have this amazing 14. We have the double head. So these are more abnormalities, okay, of the sperm. So human sperm move at speed of about 3 millimeter per minute through the female genital tract, and sperm reach the uterine tubes 30 to 60 minutes after copulation. Contractions of the organs may facilitate the transport of the sperm to the uterine tubes. And that will be that ends our lecture for today. Okay. So this is I said this is just a short topic. So just to cover our topic for the prelim exam. Okay. So our topic, we will be taking the prelim exam based on our discussion. Okay. So we have from our lecture one, lecture two, and then this lecture. Okay. So this comprise the male reproductive system. Okay, thank you.